a few cores from the Neurotechnology Center will be presenting today. So I'll start by talking about the Optogenetics and Neural Engineering core, and then we'll hear from the Idea core and Machine Shop. All of us work pretty closely together, and there's some nice synergy between the cores. Okay, so I'll start by describing what we do. Our overall mission is to enhance research on the Anschutz Medical Campus, and we do that by providing in-house engineering support for your research. So I think really the key takeaway for this entire presentation is that we provide engineering support for your research. Okay. So how do we do that? Well, our goal is to reduce the barriers to entry for you to do new experiments or to do new analyses, to really try to take your research to the next level. A little bit about our history that will also kind of serve as an example of the kind of things that we do. So we're focused on neuroscience. That's what my lab does research in. And I started a version of this core several years ago. But also we are supported right now by the Neurotechnology Center. But what we do is really applicable to a lot of other types of research. It really is not limited to neuroscience. So we started in 2012 with internal funding from the Dean's Office via the Strategic Infrastructure Research Committee, or CIRC, that some of you are probably familiar with. And we actually started with a focus on optogenetics, right? And that's what the O in one core stands for. Optogenetics this is something I probably wouldn't have to explain to a specific neuroscience audience, but I'll just give a very brief explanation in case it's not familiar to everybody. It's a set of methods that allows cellular processes to be controlled with light. And this has been very big in neuroscience over the last decade and a half or so, because it allows us to control the activity of specific types of neurons with light. And light has really good spatial and temporal resolution, right? So in a lot of our experiments, we want to manipulate neural activity and see how that affects maybe processing in some other part of the brain or maybe even behavior. And these are methods, those are goals that neuroscientists have had for a long time, and there's a lot of other ways to manipulate activity. But there's some very specific key advantages of using this technique of optogenetics. Specifically, we can manipulate activity of specific genetically defined cell types. That's where the genetic comes in. And again, we can do it with really high temporal and spatial resolution, much more so than, say, drugs, right, which act relatively slowly. So, so initially, we were focused on helping labs, and neuroscience labs, but also other labs that were interested, incorporate optogenetics into their research. And I'm kind of telling you all this to give you an example of how we try to reduce barriers to getting the research to the next level. So this is a technique that arose in neuroscience the first paper was in 2005, and it really started taking off a couple of years after that. And so a lot of neuroscience labs were in a position where they could be doing better research if they could harness the power of this approach, this optogenetics approach. But it required a lot of specialized equipment, light sources like lasers or LEDs, the know-how of using these effectively, how to analyze the data, how to troubleshoot. And that's not something that most neuroscientists were trained to do because it's a pretty new technique. So our goal was to make it easier for labs to start adopting that kind of technology. And so that's how we started in 2012. Again, I want to emphasize that optogenetics is not specific to neuroscience. You can control many other cellular processes, not just neural activity with light. And there are many labs on campus that are developing those sorts of techniques and using those techniques. In 2015, we became one of the cores as part of the renewal of a P30 grant from the National Institutes of Health, and specifically neurological disorders and stroke. So again, there's an N there, neurological. So we kind of became more focused on the neuroscience community while still being able to help other labs as well across campus. And since 2019, we've been part of the Neurotechnology Center. again, focused on neuroscience, but not limited to neuroscience. And so, I'd say that we're more focused on the neural engineering aspect of what we do now, rather than specifically the optogenetics. But in all of these cases, what our goal is to help get your research, labs research on campus, et cetera, to the next level, right? We're all doing, obviously we're all doing great research, but we probably know that we can be doing even better research, and sometimes it's helpful to have that type of support. And specifically, engineering support, right? Many of us, so this will sort of echo what I said about optogenetics before. Many of us biologists have a particular kind of training that might not have so much of a quantitative or engineering related background in it. What I always say is that if I could clone myself, I'm a systems neuroscientist, I came from a psychology background. If I could clone myself, I would be an engineer so that I could do even better research. And so instead of inventing the technology to clone myself, I started this core so that we'd have engineering support. And that's what we've been doing for the last number of years. And yeah, the goal is really to provide that kind of support for labs that need a little bit of extra help to take the research to the next level. So a few of the specific things that we do, just to give you some ideas, we provide support, we provide optics related support. So this of course relates to the optogenetics experiments that I was talking about before. So we have various light sources like lasers that you can rent. We can design patch cords to get light from your light source into the type of tissue you want to get it into. And we can quantify, we can characterize the light sources like your lasers. When you buy a laser off the shelf, you can look up the specs, and we have found empirically that those are typically not actually what the laser is producing. So it's pretty important to quantify what the laser is actually doing and how to drive it in a way that will allow you to interpret your data. Okay, so we provide support in optics. We provide electronic support. In neuroscience, there's a lot of need for microcontrollers that can kind of control behavioral experiments in real time. And that has gotten us into providing support in electronics in general. So you kind of think of us as like an electronics bench or an electronics workshop. So if you need to incorporate some of that kind of thing into your research, please talk to us. We do some work with software. We can analyze data, talk about data storage, make your software more user-friendly, etc. And finally, we do a decent amount of 3D printing. And I'll talk more about this on the next slide. But the key point here that I want to convey is that we try to be kind of a jack of all trades. So we try to provide engineering support in all of these realms that can help labs with the research. I guess I should say, if you guys have questions along the way, just feel free to unmute and ask, or put something in the chat. I have the chat box up, so I should be able to see it if you drop a question in the chat. Okay, so I thought I would talk about our 3D printing in a little bit more detail, because I think that's most applicable to like the widest range of labs on campus. So we have a 3D printer there's a number of types of 3D, excuse me, of 3D printers. We'll see them on this slide. We have a fused deposition modeling printer. This allows you to print very flexibly, print many objects very flexibly, fairly inexpensively, using a wide range of materials. So this is just an example showing a little holder here that we've printed for a particular type of experiment. There are various other types of printers that we don't have in the lab, but we have access to. So stereolithography, which allows for higher resolution printing with watertight materials. It's bio-safe, biocompatible. Polyjet printing is another complementary type that allows for multiple properties in the same print, like different materials or different colors. And finally, quite recently, we've made some contact with a lab in Boulder, a core lab at CU Boulder that has a nanoscribe printer. And so this does super high resolution using pretty cool technology, 2-photon polymerization. So this is a CU icon that we printed at a very small scale that you can see here. So I want to convey that it's not, okay, the way we interact with you and printing is if you need to get something printed, you can come talk to us and we can help you design the files that will get it printed. And in some cases, if it would be appropriate to use this FDM printer, we can print it for you. Okay? In other cases, we can advise you on which type of printer would be best to use for your needs. And we can kind of outsource it to other printers off campus that we have relationships with that have these types of printers. And again, this nanoscribe printer is a pretty new development. Andrew Skellen is the manager of the OneCore. So you'll see him on the last slide. And so he's been up to Boulder recently and has been trained on this printer, so he can actually get some very small things printed. Okay, I have a question. Could you 3D print some devices for animal handling, for example, mouse head holding devices for our 9.4T MRI scanner? I'd say the answer is yes. We've printed a bunch of head holders already for animals, like for head fixed type experiments. So we'd of course have to talk about the details of how that would be, what you would need for the scanner. But we don't, I don't think that the materials that we print would be a problem in a scanner itself. They're not magnetic, they're not metal. And yeah, there's just a lot of ways to print these kinds of things. So yes, Natalie, you should talk to us about printing something that would fit your needs. Yeah.
Thank you so much. Thank you.
Okay, so again, we can either print something for you right here on campus, or we can kind of serve. I guess this is we can serve as a go-between. Let's just say that we can facilitate you getting something printed in the right way, even if we don't have that particular printer here on campus. Okay, I thought I'd also just give a few examples of some of the resources that we support on campus. These are going to be pretty neuroscience centric and neuroscience heavy because that is what we focus on. But again, these are just sort of examples that give you some idea of what we can do. And these are really, these examples are driven by the needs that have been brought to us by users. So if there are people on campus who, are doing something outside of neuroscience, but would like some support in that particular type of experiment or using that type of open source resource, we're happy to help with that. So one example is software called Deep Lab Cut, which is really useful for tracking animal behavior. So here's a mouse that's poking around in a box, performing a particular task, and using a deep learning algorithm, we can track different features on the mouse that you can see with these colored dots here. And so this is something that's just really useful for a lot of neuroscience experiments. It can be applied to real-time experiments, like maybe you want to deliver some kind of stimulation when the animal is in a certain place. So you can be tracking the animal in real time and deliver that stimulation when you need to within your behavioral system. This was not something that we developed. I just want to be clear about that. This was developed by Mackenzie Mathis, and it's open source though. It's designed for anybody to use. So strictly speaking, it's not like you need to work through the OneCore to use Deep Lab Cut, but there's a decent amount of overhead in getting it set up on the computer and getting these networks set up so that you can effectively track your animals. And we can help reduce that barrier to using this kind of technology. Okay, similarly, we've offered a lot of support for the use of Miniscopes. So here's a Miniscope implanted on a mouse. And what these are small, lightweight 1-photon microscopes that allow for imaging neural activity. So you might express a particular fluorescent marker that fluoresces when a neuron is active, like GCamp is one of the more commonly used ones. It's GCamp 6F. And these are expressed in a particular type of neuron, glutamate neurons in the pallidum in the brain. And so what you're seeing here, these little kind of blinking lights, is basically a proxy for activity of the neuron. You're actually seeing calcium concentration. And so this allows you to record the activity of neurons of a particular type, and not just one neuron, but you know, all of the neurons that happen to be in your field of view. Okay, so again, this is pretty specific for neuroscience, but you know, you can imagine similar types of experiments in other fields that might use a similar type of technology, and we would be happy to support that. Okay, and okay, so a couple of questions. First of all, Andrew, thank you for replying more to Natalie. Mike Meshes asks, how big is the field of view typically for the Miniscope? I'm looking off screen for neurons. Yeah, I mean, it's a good question, Mike. You could see hundreds of neurons. There are different, there's Miniscope imaging. You know, there's also mesoscope imaging, which of course is different from Miniscopes, and that's not implanted. I'm gonna say it's on the order of like a millimeter or so, millimeter diameter. Does that sound about right to you, Ryan?
I think maybe 200 microns.
Okay, less than a millimeter, maybe 200 microns or so. Yeah, it's possible that, oh, Andrew, okay, Andrew's already answered that. Good. There's a new scope V4 with 1 millimeter diameter field of view. Okay, so maybe that's the maximum. Cool. All right, and then another recording technology that we've started supporting is neuropixels. So this is a type of electrophysiological probe that has many, many closely packed contacts on it so that you can record from a bunch of neurons in the brain at the same time at the highest temporal resolution available. So this is higher resolution than Miniscope imaging. This is electrophysiology, so you're recording the electrical signals directly. And this, again, you know, all of these technologies were developed off campus, but they're all open source, and the intention of the developers is for them to be used as widely as possible. And yet, it's not trivial to just start using a new technology like this. So the OneCore can help you get that set up in your lab. In the case of NeuroPixels, we actually have a data acquisition station that you can rent and use with the probes that you buy individually. So you can acquire the data and do the first pre-processing steps on the data as well. So again, these are just examples of how we can reduce the barrier to labs using new technology and making their research better. Okay, of course, we do have to charge something for this because we're a core. We have the lowest rates for members of the Neurotechnology Center. Membership of the NTC is at the department level. So if your lab, or the lab that you are working with, is a member, if the lab is part of a department that's a member of the NTC, you get the lowest rates. So if you feel like you're going to be using NTC services, you might want to convince the chair of your department to become a member of the NTC. And you could talk with Mark Delacqua, the director of the NTC, about that. But we also, of course, work with other labs on campus, and we even work with off-campus labs, just at slightly different rates. We don't want cost to be a barrier to you using our services. That would be kind of self-defeating, right, if we were too expensive for people on campus to use. So feel free to talk to us about what we can do for you, and we consult on what we can do for free. And we can also give you an estimate, of course, before you get locked into paying a lot of money for something that maybe it turned out you didn't want. And again, like this one, none of this would really work if we were charging too much money, right? This is supposed to be a service. We're not trying to make a profit here. Okay. All right. So if you want more information, you can email us. This is my email address. I'm the director of this core. Andrew Scallon, as I mentioned, is the core manager. He's already been answering some of the questions on the chat. You can email him at Neural Engineering, CU Anschutz. We have a lot of information on our website. I see that Andrew actually already dropped a link, I think, to that in the chat. Maybe you didn't drop a link. He mentioned it though. You can. So here's our shortened website, Big OneCore. Again, we're part of the Neurotechnology Center. We are currently located on the 7th floor of RC1 North. in the lab space there. We are in the process of moving to the first floor of RC1 North, specifically room 1402. So this is one of the multi-purpose labs. So please contact us if you have any questions.